All right, so this lecture is on chapter 18, just the first section, so 18.1. Just talking a little bit about DNA damage and mutations. So DNA damage happens all the time. About 1 million times a day, your cells have some DNA damage. Can be physical changes. It can be mismatches that haven't yet been repaired. The point of a DNA damage, or the difference between a DNA damage and a DNA mutation, is this stuff is repairable. Okay, so this is all happening as the cell's going on in G1 phase, okay, or G0 if the cell's not getting ready to divide. When it becomes a mutation, and that's what we're going to focus on, is when you have a change in the base sequence. It cannot be repaired because this is post-replication, which means that that mutation, that change, maybe you had AAATGC, and you get a mutation. Okay, I'm only writing one strand of the DNA here, TGC, and that gets replicated. Now, so I'm going to show you that's where the mutation is. Now your strands 5' ACTGC and your 3' TGACG look fine to the cell, right? You got all nice base pairing happening. And once it's been replicated, it can no longer be repaired because it's not recognized by the repair mechanisms. So DNA damage, again, happens all the time. You have all of these repair DNA polymerases, so lots of polymerases that are important for recognizing different types of DNA damage and fixing them. We unfortunately didn't get a chance to go through this semester. When it goes through replication with our delta epsilon and becomes permanent, that's what we call a mutation. So that's what we're going to focus on, mutations, changes in the DNA sequence. Now, you can have somatic cell, which is all the cells of your body except the germline or the germline or producing cells. So these are the ones that produce gametes. So a somatic cell mutation is just going to affect you. It's not going to affect the new or the next generation. A mutation in your germline, so your egg or sperm producing cells, are going to make mutations in the gametes. And it's kind of sad to call your babies mutants, but I kind of think it's funny too, just because, I don't know, who would call their mutant progeny? But you can understand that it's affecting the next generation. Okay, this is just another illustration of the same idea. So a germline mutation, say in the sperm here, is going to affect this entire organism, this whole next generation, but only half of the gametes, right? Because they still have a wild type or non-mutated, in this case, from the mom. Okay. Somatic cell mutations, again, are just going to affect you. They're not going to affect the next generation because they're not in the reproductive tissue. And there's, a kind of a negative connotation with the term mutation. That's why I also think it's kind of funny to call it a mutant progeny, because perhaps this progeny is, now got a very special feature to them. So remember that mutations are not all bad. Mutations are the source of genetic diversity and evolution. So without changes in gene sequence, there's no way to adapt and evolve with the environment. We also make mutations in the lab on purpose in order to study gene function. So let's just talk about a couple different types of mutations. So you could get an error in replication that is continued on to the next DNA post-replication, and one is called a base substitution, where I want you to see that you just put in a different nucleotide. Okay, so instead of a T that was in the original sequence, now there is a C. And we've talked about our codons, and so if this was in a protein coding region, you've changed one codon. When you have a base substitution, we name them either transitions or transversions. So transition is substituting a pyrimidine to a pyrimidine or a purine to a purine. Okay? And these are tolerated a little bit more than transversions because you can see that you still have the kind of the same size for the base pairs. So you've removed an A purine and it's been replaced with a G. It's still going to fit in that double helix, though the base pairing won't be ideal. Whereas a transversion, where you're going from a purine to a pyrimidine, so in this case you have a lot more options, right? A can be either C or T. Likewise, C could be either A or G. These are actually usually picked up by the repair mechanisms because you've either got something that's making the DNA too bulky or too skinny compared to the normal pyrimidine-purine base pairing. So if you see the word transition, you know it's purine to purine. Transversion is purine to pyrimidine. Okay. And so what can the consequences be if we're affecting just that one codon? So we'd have three different types: missense, nonsense, and silent. Missense means you've changed the codon and you're getting a new amino acid, right? So in here, we normally had TTA, here we had normally TCA, which encoded for serine. It's now been mutated to TTA, which is the codon UUA. So top strand is always the coding strand, bottom is always the non-coding or template. And now you get leucine. So you've changed the amino acid. That's called a missense. You can also get a change where you add in a stop codon, and that's called a nonsense. So it was an amino acid, now you've terminated translation. That's a nonsense mutation. Or, like we talked about, due to the redundancy in the genetic code, you might have a base substitution, but you end up with the same amino acid. And that type of mutation is called silent. Okay. So again, you will use your amino acid codon table to figure this all out. There's also something called a neutral mutation. So this is when you have a missense mutation, but the amino acids have similar properties. I was talking about this a little bit in class. And so you might have a change in a codon that gives you a missense, and glycine is now alanine. Okay. These amino acids are very similar. Remember, an amino acid, it's the R group. It gives the amino acid its property. The rest that's not highlighted is the N-terminal, C-terminal, or amino, carboxyl, ends, and hydrogen. So this backbone is the same in all amino acids. And so substituting an alanine for a glycine may not change the protein structure. Protein might retain its function. So in that case, the missense is called neutral. When you're having a substitution that doesn't change the protein structure, the protein function. Now, we don't have enough data to determine this, but I want you to understand where this idea comes from. And again, if you go and look at the mRNA codon table, you can see that the codons for glycine, where are we? Glycine is G, G, and then I'm going to put N because it could be U, C, A, or G. And the codon for alanine is G, C, U. And the only difference is right here. So if you have a mutation in this codon in the third nucleotide, you're still going to get glycine. If you have a mutation in the second, this should be N, you could get alanine, which is very similar. So again, we think that some of the evolution and the redundancy of this code allowed for these base substitutions to not be totally detrimental. If you changed that first codon, G, from glycine to a C, CGN is arginine. And arginine, where is arginine? Is a charged amino acid. So that's going to have a much bigger impact on the protein. The other type of mutation you can have is an insertion or a deletion. Okay, again, due to an error in DNA replication. This is going to change the reading frame, right? Because our reading frame is groups of 3, and if you add something in or you remove something, your groups of 3 change. So if your groups of 3 change, you have a reading frame mutation, which means everything downstream of that is going to have a different amino acid sequence. Okay. How this occurs is it's proposed that you can have what we call a wrinkle in the DNA where either the replicating strand wrinkles a little bit and you kind of get off and you add in an extra nucleotide, or the template strand gets a little wrinkled and you lose a nucleotide. And there's a little video here you can watch about kind of how they think this might happen. Okay, so these are just the next couple slides are just examples of insertion deletion consequences. Again, changing that reading frame. So here you added an A, and that changes the groups of 3. Here you lost a C, and that changed the group of 3. And in this case, gave you a premature stop codon. The only time an insertion deletion doesn't have a frame shift effect is if you insert or delete in a group of 3. So if you insert or delete group of 3, a whole codon, it's called an in-frame mutation. Okay. And honestly, I have no idea how common that is. So let's do a couple, which would have been clickers. I suggest that you take a look at this, pause the video, figure out the answer, and then come back. So in this example, we need to go and find our ATG grouping or our reading frame, sorry. And what we have is a base substitution from an A, now a U, and again, we're always going to write the coding strand so that we don't have to base pair and go anti-parallel. CAC encodes for histidine. CUC encodes for proline. So this would be a missense mutation. Here we have another group, ATG, CAC, so CAC, oh sorry, go ahead and pause, come back. CAC, again, codes for histidine. CAU, if you look at your chart, also encodes for histidine. So we call that a silent mutation. Okay. Here's a one that I want you to, it's going to take you a minute to think about. What if a transition occurs, a single transition in the codon CAU? What amino acids could be specified? So go ahead and pause the video, do the work, and come back. So a transition is purine to purine, pyrimidine to pyrimidine. So the first transition we could have is C becomes U, right, because there's no T's. And CAU encodes for histidine, and now we have tyrosine. Or you could have a transition at the second nucleotide. So no, that's not a transition. CGU, which encodes for arginine, or you could have a transition at the third nucleotide. Why am I doing this? I'm base pairing. I'm not doing my, okay, this is going to take some brain power. U to C, CAC, which also, or which, yeah, that would be silent here, encodes histidine. So this is the correct answer. Okay, here's another one. What would happen to the protein produced if the DNA strand was mutated as shown? Go ahead and pause the video. So what we can see is that we have a deletion. A deletion is always going to result in a frame shift. So an insertion or deletion is going to result in a frame shift and no more analysis. I don't want you to keep going and looking for nonsense or missense or silent mutations. Once you see frame shift, you are done. Okay, and this is just showing you the frame shift. Oh yeah, so this is 1, 2, 3. This is the fourth amino acid. 1, 2, 3, 4. Right. What would happen with this one? Go ahead and pause the video. So now we have a UAA, which is a stop codon. So we call that a nonsense mutation. Okay. So practice some of those. You'll have a bunch on your recitation worksheet. One other type of mutation I just want to talk to you about is nucleotide repeat expansions. Some of you have done genetics in the news on these types of mutations. So a nucleotide repeat is where you've got the same, and you can see here it's GTC, repeated over and over and over again. And there are genes that have these natural repeats. Well, sometimes during DNA replication, the DNA polymerase gets a little lost in where it's replicating. And you can get this hairpin formed. And because it's this repetitive sequence, the DNA that's being synthesized realigns somewhere, and you're going to end up with all of this extra DNA. So you're going to have an expansion of that repeat sequence. End result is you started off with what? 1, 2, 3, 4, 5, 6, 7, 8 repeats, and now you've got like 13 repeats. Okay. So this issue is called slippage, and we don't always understand exactly why it happens, but it does happen in some of these diseases, as shown in this figure. And we barely mentioned this word anticipation. So what happens with these diseases is we call it anticipation, whereas there's this potential for the next generation to have an increased number of repeats if you get slippage in the germline cells. Okay. So Huntington disease is a really well studied one, and what this is showing you is that normally people will have between 9 and 37 copies of this CAG repeat. But for some reason, when you get to 37 or above, and sometimes these numbers overlap, and sometimes these numbers have a missing numbers, it's just the data we have at the time. When you get up here, you start to see disease. And many times, the more repeats you have, the more severe the disease is. So my dad's girlfriend has a type of muscular dystrophy. And it's a really interesting type because it only affects the eyes and the throat. So it's a trinucleotide repeat disease, autosomal dominant inheritance, and muscular dystrophy results in loss of muscle. So she's actually had several surgeries to kind of pull up her eyelids because she doesn't have the muscles to keep them open. Unfortunately, she can barely swallow, so she's had a feeding tube for years now. And the worst is that her muscles are starting to atrophy in her legs, and so she's having a hard time walking. And she's the first one in her family to actually get a genetic diagnosis where they did sequencing of her DNA. But she said if you go back and look at her pictures of her family, through the generations, you see these droopy eyelids. So it's been something that's been passed down. Her mom basically starved to death. She didn't know what was going on, but she couldn't swallow anymore. She couldn't eat. So anticipation means each generation it can get worse. It can stay the same. My dad's girlfriend never had any children. And her brother was tested, and he does not carry the trinucleotide repeat sequence that's near the disease kind of limit there. So his children are all fine. So this is just showing you what can happen with this idea of anticipation. So as people... in each generation you get an increase in the nucleotide repeats, the age of onset gets earlier, and the manifestations or the severity gets earlier. And this is just a pedigree that's showing the age of onset and the number of repeats. And so they didn't have the data on the number of repeats here, but you can see that the age of onset starts getting younger and younger, and the repeat numbers are increasing. So the more the repeats, the younger the age of onset. So, final